Everyone, and thank you for joining us this week for Research Innovation. We only have one speaker today, Dr. Sue Gu. Dr. Gu did her undergrad at Yale University, followed by her MD at Columbia, and I'm gonna put my glasses on so I can actually read this. And then she came here to do her internship, residency, and fellowship. She is currently a clinical instructor in the pulmonary division, and we'll be talking about macrophages in the right ventricle.
Thank you so much. I'm excited to be here today to talk to you about some of my favorite subjects. So my objectives for today are first, to establish the importance of the right ventricle in pulmonary hypertension. Secondly, to tell you a little bit about how I characterize macrophages and their role in the RV and pulmonary hypertension. And third, to talk about some of our future directions, which is determining the contribution of complement activation of RV macrophages to RV failure and pulmonary hypertension. All right, and is everybody on Zoom hearing me okay? Okay, great. So beginning with one of my favorite subjects, which is the right ventricle in pulmonary hypertension. I am a pulmonologist, and what I treat in my clinic is pulmonary arterial hypertension. The definition of PAH has changed a little bit recently. Most of you might know that by definition, the mean pulmonary arterial pressure of greater than 20 millimeters of mercury is required, as well as a pulmonary capillary wedge pressure, which essentially rules out possible left-sided heart disease of less than 15 millimeters of mercury. And then a little bit of changes and a little controversy lately is depending on which definition or guidelines you look at. The pulmonary vascular resistance for PAH is defined as either greater than 2 Woods units, which is according to the most recent 2022 European Respiratory Society and European Society of Cardiology guidelines, or greater than 3 Woods units, which is what we've been using for a long time. Most recently was updated in 2018 by the World Symposium on Pulmonary Hypertension. So not quite universal agreement on that yet, but in general, these are the definitions of pulmonary arterial hypertension. And although we've made a lot of progress over the last many decades in treatment of pulmonary arterial hypertension, it does remain a disease with very high mortality. So one of the biggest cohorts that's been studied of PAH patients is the REVEAL cohort. And in an analysis of the cohort from 2006 to 2009, the median survival was still at around 7 years, with mortality at around 3 years from diagnosis at less than 70%. Since that time, we have made some strides in PAH therapy, and the more recent FAR registry that just came out, analysis of a registry that enrolled and is still enrolling from 2015 through the 2020 cohort, still shows that mortality remains high at 3 years, but at closer to 80%. So we've made some progress, and that's mainly due to a lot of FDA-approved treatments for pulmonary arterial hypertension, targeting different pathways, such as the endothelin pathway with ambrisentan, which is an endothelin receptor antagonist, other endothelin receptor antagonists like bosentan and macitentan, the nitric oxide pathway, which is targeting the PDE5 inhibitor pathway, sildenafil or sildenafil, or direct guanylyl cyclase stimulators like riociguat, and then the prostacyclin pathway, which has been around for the longest, with IV prostacyclin therapy available for several decades now. And this leads directly to increase in cyclic AMP, leading to vasodilation and antiproliferation. But what makes all of these treatments, what they all have in common is that they're all treatment of the pulmonary vascular bed and the pulmonary vascular resistance. But mortality in PAH correlates with RV function and not the PVR. So in this study, which followed prospectively patients that were diagnosed with PAH and then started on PAH vasodilator therapy, you can see that if you follow the patients that survived, there's actually no difference between survivors and non-survivors and whether their PVR changed. But there is a significant difference between survivors and non-survivors and changes in RV ejection fraction. So in patients that had a decreased RV ejection fraction, they were more likely to have mortality. And you can see pretty dramatically that patients, even patients that had a reduction in PVR and a decrease in the mean PA pressure, some of those patients could still develop decreased RV ejection fraction and RV failure. And there's this very steep drop-off curve in survival for those patients. And then just stepping back, I've been talking about pulmonary arterial hypertension, but let's not forget that pulmonary hypertension as a disease category encompasses not just this relatively rare form of pulmonary hypertension, which is PAH, which is defined as WHO group 1, but also the much more common causes of pulmonary hypertension, especially in the United States, which is pulmonary hypertension due to left heart disease, pulmonary hypertension due to chronic lung diseases, and then pulmonary hypertension due to chronic blood clots or chronic thromboembolic pulmonary hypertension, and then group 5, which is PH due to unclear or multifactorial reasons. So the reason I say that is that all of these different diseases all lead to one common pathway. So all roads lead to Rome, or the right ventricle. So right ventricular failure and all of those forms of pulmonary hypertension, not just PAH, is predictive of mortality. And that includes people admitted to the ICU with acute right ventricular failure, whether it's from an acute PE or sepsis or acute MI. And the progression of right ventricular failure and pulmonary hypertension, from what we understand, is that in a normal state of hemodynamics, the RV is normally a smaller, thinner walled structure compared to the LV, which is a bigger size and wall thickness. And then in early pulmonary hypertension and in a relatively compensated state, the RV undergoes adaptation where the RV wall might increase in thickness and the RV contractility increases, allowing for the RV and the pulmonary artery load to remain coupled. Some of these processes that we understand are happening are compensatory angiogenesis and then cardiac myocyte hypertrophy. But then in late pulmonary hypertension, for unclear reasons, some people develop this switch in which there's maladaptive changes that occur where the right atrium begins to dilate and the central venous pressure therefore also begins to increase. The right ventricle dilates in size and diameter, and the wall becomes thin, and then the size of the right ventricle actually starts to push into the left ventricle, and you get septal bowing, and the left ventricular cavity gets smaller in size. And on a molecular and hormonal level, there's a number of changes that we know are going on in the failing of the maladaptive right ventricle. So there's apoptosis of cells, there's metabolic reprogramming, there is capillary rarefaction where there's not enough capillaries potentially developing substrate or delivering substrate to the hypertrophied right ventricle. There's reactive oxygen species and oxidative stress, fibrosis, neurohormonal and RAS dysregulation. And then what I'm interested in, which is inflammation and immune cell recruitment, is happening in the right ventricle, and little is known about why or what exactly that is contributing or causing to right ventricular maladaptation. So in patients that have this state of chronic heart failure, they're very easily pushed over by precipitating factors, whether it's infection, pulmonary embolism, a bleeding event or bleeding disorders, or just progression of their disease into an acute decompensation. And these patients are often our sickest patients in the ICU. They're deteriorating, they become hypotensive systemically, which then worsens the blood flow to the coronary arteries, which perpetuates the cycle of ischemia, might lead to arrhythmias, which leads to further hypotension. So many patients get stuck in this spiraling circle of death, of acute RV failure, and mortality from this condition can be as high as 40%. And if we're able to stabilize them through a mixture of inotropy or diuretics or pulmonary vasodilator therapy, some patients are able to stabilize and discharge, but mortality even in the post-discharge period can be as high as 35%. So that's all to say we don't have treatments that are targeting the right ventricle, either in chronic right heart failure or in these acute instances of acute deterioration. So that brings me to my research interest, which is macrophages in the right ventricle. And this is a stain of the macrophages through a thick slice of heart for CD68, which is a pan-macrophage marker. And what we understand is that macrophages are the major immune cell of the heart. The makeup of the heart is primarily predominantly endothelial cells, followed by cardiomyocytes. There's also a population of fibroblasts and pericytes and other mesenchymal cells. And immune cells only make up about 6% of the cardiac composition of the heart. But of that, at steady state, macrophages and dendritic cells make up the vast majority of myeloid or immune cells in the heart. And what we know that they are doing in the heart, just like in all tissues of the body, macrophages play really important roles in tissue homeostasis and maintaining tissue function. Resident macrophages in the heart are doing some really important functions as well. So, for instance, they are known to facilitate electrical conduction in the heart, and macrophages sit near this AV bundle, which is where the conduction is going through from the atrium into the ventricles, and that the macrophages, if you deplete or remove the resident macrophages, you actually develop complete heart block. So in the absence of these macrophages, these connections and gap junctions that allow the conduction to go through the AV bundle actually completely stops or goes away. Other things that we know about what resident macrophages are doing in the heart is that in the homeostatic heart, the cardiac myocyte, because it's undergoing so much, basically because it's beating constantly and constantly metabolizing different substrates, mitochondria are getting used up, and they're old mitochondria that are produced as byproducts of just normal homeostasis. And again, resident macrophages, which all express this receptor of CX3CR1, these resident macrophages recognize these kind of apoptotic bodies and debris that are tagged for engulfment. And they're professional phagocytes that then engulf the debris that's produced by the normal homeostatic heart. And in the absence of these resident macrophages, again, if you deplete them, then you get all of the cellular debris and old mitochondria that are not cleared, and that leads to myocyte dysfunction. And then one more example that gets to the nuances and how difficult it might be to figure out exactly what macrophages are doing at different stages of disease. From the studies of myocardial injury and myocardial ischemia, we know that recruited macrophages, which express CCR2 positive and are coming from the bone marrow, they're called into the heart, cross over through the blood vessels into the heart tissue. And if they're not present at the early stages of myocardial injury, then the heart can't respond to that acute stressor, and you can actually get myocardial rupture. But then if these cardiac macrophages persist over a long period of time, these kind of pro-inflammatory, acutely recruited macrophages, that can lead to heart dysfunction. So a prolonged amount of inflammation is not helpful in case of repair for myocardial injury. And what's necessary, it seems, is a switch to more of this reparative tissue resident macrophage, which is these CCR2 negative macrophages, to turn off the ongoing inflammation and to kind of switch the programming of the tissue medium back to a more reparative phenotype. And so these recruited CCR2 macrophages that I mentioned are also required for acute changes in afterload in the left ventricle. So in a mouse model of transaortic constriction, where a band is placed around the aortic vessel, in this early pressure overload period, if you start giving an antagonist to CCR2, then these hearts actually don't develop the features of late stage pressure overload and heart failure, like systolic dysfunction, hypertrophy, and fibrosis. So back to the right heart and how I'm studying right ventricle macrophages. These are the experimental models that I've used throughout my research. So these first three are mouse models of pulmonary hypertension: hypobaric hypoxia, schistosomiasis, which is actually the most common cause of group 1 PAH outside of the developed world. And then a bleomycin intratracheal instillation is another model for a type of group 3 pulmonary hypertension, where it causes pulmonary fibrosis as well as pulmonary hypertension. And then the rat model of Sugen hypoxia, which out of these is the most severe and results in the most right ventricular changes. So starting with the first model of hypobaric hypoxia, does anyone recognize what this mountain is?
I think it's Everest. No? No, it's actually Evans.
A little lower.
It's Evans, isn't it?
So this is Mount Kilimanjaro,
Oh,
which is 19,000 feet. I put this as reference because this is about where we're putting our animals when we put them in a hypobaric hypoxia chamber. So we depressurize the chamber to simulate an elevation of 18,000 feet, at which they're breathing about 10% FiO2, which is about half of the room air FiO2 that sea level people are breathing. Since we're in Denver, we're actually breathing a little less than 21% as well. So in hypobaric hypoxia, in our mouse models, this results in some mild pulmonary hypertension and mild RV remodeling. So in our hands, at 3 weeks, which is kind of considered a more chronic hypoxia exposure, we get elevations in the right ventricular systolic pressure to about the 35 to 40 range, which is above the kind of normoxia range of the normal RVSP of 25. And then there's some changes in the right ventricular mass, which we normalize to the total size of the heart by dividing by the left ventricular mass plus the septum. So again, some mice develop a lot of hypertrophy, some kind of a mild degree. So again, this is kind of our mild pulmonary hypertension experimental model, and it's easier to study because it's a mouse model and we can get genetic variants of mice and reporter mice. What I've learned from studying mouse models of pulmonary hypertension is since flow cytometry and antibodies are so readily available for mice, is that I've been able to identify a number of different right ventricular macrophage populations that match with what's been reported in the literature of other subtypes of macrophages. So in this is kind of one of my earlier studies. In this earlier example, I excluded, in general flow cytometry plots, you exclude debris, you exclude doublets and only look at singlet cells. You exclude dead cells, and so of this fraction that's remaining of a population that expresses CD45, which is present on all myeloid cells, CD11B, which is common to macrophages and dendritic cells, and then an even more specific macrophage markers, which are MERTK, which recognizes that apoptotic label that's on dead or opsonized cells. which is MerTK, and then CD64, that gating based on expression of CD11C as well as MHC2, which is involved in antigen presentation. You can identify 3 what are called interstitial populations of macrophages, or IM1, 2, and 3. And this has been mostly reported in the lung literature, but it's also been kind of validated across different organ systems. And so in my earlier studies, I looked at how acute hypoxia acutely increases the IM1 and IM3 macrophage subtypes. So at these early time points of 24, 48, and 7 hours, or 24 hours, 48 hours, and 7 days, although the overall numbers of macrophages are kind of staying stable, there's this steady rise in this IM1 population, as well as this IM3 population by 48 hours and 7 days. And when you look at the programming of the IM1s and IM3s and compare them to monocytes, these are actually the most similar macrophages to monocytes, and they express CCR2. So these are most likely getting recruited from the bone marrow and from the circulation. And then the next thing I've looked at is how chronic hypoxia increases RV macrophages. And so in these studies, I've started to separate, I started to look at both the left ventricle and the right ventricle as an interesting comparison, because, you know, both parts of the heart are subjected to the same stimulus of the hypoxia, but only the right heart is seeing that acute pressure increase and the increase in the overload. So I'm using the left heart almost as a control for what's going on in the right heart, just to see, isolate what's going on in response to pressure overload. So here I'm showing that at, compared to normoxia, that at 3 weeks, the number of macrophages per cells per milligrams of tissue in the right ventricle is increasing in the RV. And I can then further identify this CCR2 positive macrophage population by flow cytometry. And I've used both antibodies as well as reporter mice that express CX3CR1, which is that resident macrophage population in a green fluorescent marker, and then CCR2, which is this recruited macrophage population in a red fluorescent marker. So using both techniques of antibody and reporter mice, I've done time courses of chronic hypoxia exposure. And what I've seen is that chronic hypoxia pH significantly increases the CCR2 macrophages in the right ventricle by 3 weeks. So again, using the control or the comparison to the left ventricle, I think what's most striking is that at 3 weeks in the left ventricle, the CCR2 macrophage population is staying relatively stable in terms of density, normalized to the milligrams of tissue, whereas the expansion is pretty significant in the right ventricular tissue. So moving on to other studies I've done with bleomycin pulmonary hypertension. This is an animal model for group 3 pulmonary hypertension, like I said. And there is some mild pulmonary vascular remodeling, some modest RVSP elevation, and there is some RV fibrosis that happens in this animal model. So this is an example from some published studies of the RV peak systolic pressure, which is a little bit higher than what we saw in the chronic pulmonary hypertension model. So RVSP in the 40s. And then a little bit more marked change in the RV to LV plus septum ratio. And then in these mice, looking at the cardiac output was decreased in the bleomycin treated animals. So these animals, again, they get bleomycin intratracheally so that it's basically directed only at the lungs and causes the fibrosis and the changes there. And the studies, so in the studies that I've done on the bleomycin effect on pulmonary, on right ventricular and septal macrophages, again, I'm getting even more granular, and I'm dividing now both the right ventricle and the septum from the left ventricle, because as I've moved on, I've gotten more interested in kind of all these different compartments of the heart, because the septum is so close to the right ventricle, and it's being affected, as I've shown in the right ventricular failure, that it's hard to say whether the septum is closer to the left ventricle or if it's closer to the right ventricle in terms of molecular changes in inflammation. So in these studies, again, I've separated RV, LV, and the septum. What I show here is that macrophages actually, not significantly, but they go down slightly from the LV to the between the control and the bleomycin pH conditions. I think I'm missing some of my stars and p-values here, but there's a significant increase in macrophages in the bleomycin treated RV as well as in the bleomycin treated septum. And then that's in comparison to neutrophils, which there's also a decrease between the LV and the bleo and the control, and then there's this mild increase in neutrophils in the RV and the septum. So there is some inflammation there, but again, I'm missing a couple of the actual p-values here, but these are non-significant changes in the neutrophils. So in these studies with the bleomycin model, I've used another technique to characterize macrophages. That's using spectral flow cytometry. And the benefit of that is that you can take samples from these mice, dissecting out the RV, LV, and septum, and a digestion like I did with traditional flow cytometry, but you can use many more surface markers at the same time that all have a different spectral signature. And then the spectral flow cytometer will actually unmix all the signals so you don't have to do compensation and worry about overlap of signals as much, even with up to 20 or more markers at the same time. So in these studies, I did the kind of usual gating, this traditional gating to identify macrophages with CD64 and another macrophage marker of CD88. And then I looked at different cardiac macrophage markers that are listed here that identify different, both recruited as well as resident macrophages. So in the literature, CD163 as well as the markers TIM4 and the folate receptor 2 have been shown to identify different populations of resident macrophages. And what I've seen is that in these mouse models of bleomycin, you can get a really interesting separation between the TIM4 and the folate receptor 2 expressing macrophages, which is highly expressed in these red cells here. And then also a really clear separation of a CD163 population, which again is known to be kind of a resident macrophage cell marker. And then what's interesting is that other macrophage markers like LIVE1, which has been reported a lot in the lung literature, didn't show up as kind of a very large predominant population in any of my samples. And that again, CCR2 is a pretty small population, and it's kind of hard to make out like a clear separation of CCR2 marked macrophages. So the bleomycin pH overlaid on the control conditions. So here I have in the purple is the bleomycin pH mice, and then in the blue is the control mice. What you see is that there's actually a shift in the overall macrophage surface marker. So these kind of populations don't overlap with each other, and that this same shift happens in the right ventricle if you overlay the right ventricular control macrophages over on the bleomycin macrophages. And then these are, in contrast, if you combine the right ventricular and left ventricular macrophages together, they actually overlap together pretty similarly. So what these studies showed is that actually the overall condition of the bleomycin pH caused a greater change in the surface marker expression and separated different phenotypes of macrophages more than actually the location of the macrophages. So there was more similarity between right ventricular and left ventricular macrophages compared to similarity between bleomycin and control macrophages. And then one of the things that was interesting is that there was decreased CD163 expression after bleomycin pH. So in these kind of fluorescence plots, the control mice had much higher peaks of CD163, and then in the bleomycin, which is the red peak here, they have much lower expression of CD163. So there's this kind of shift and loss of this tissue resident population of the CD163 macrophages after bleomycin exposure. And so I'm going to talk about the last and kind of the more robust model of pulmonary hypertension that I've studied, and that's Sugen hypoxia. So this was established a few decades ago by people that worked here at the University of Colorado, and this is the model that most closely, or is thought to most closely recapitulate human pulmonary arterial hypertension. So in this example of distal pulmonary artery, in this control rat compared to a Sugen hypoxia treated rat, there's this proliferation of smooth muscle and von Willebrand factor. And so there's this thickening and this intimal proliferation in the distal pulmonary artery, in the small distal pulmonary arteries that create almost these obliterative lesions that are seen in human PAH. And then in the heart, in Sugen hypoxia, there's much more hypertrophy of the right ventricle, and there's also fibrosis of the right ventricle. So in comparison to the other mouse models that I've been talking about, this is sort of a more robust right ventricular failure model, and where we're hoping to see our biggest signal for changes in the right ventricle. So in our animals with Sugen hypoxia, we've been able to conduct cardiac MRIs here on the in the animal imaging core. So as you can see, this is a right ventricle of a rat. Here's a left ventricle, and this is a normal ratio of the right ventricle compared to the left ventricle in a long axis view and then a short axis view. And we can do measurements of things like RV area, and as the heart is beating, taxi, which is the measurement of right ventricular function, and right ventricular functional fractional area change. So those are measurements of RV, or all measurements and biomarkers of RV function. We can also measure kind of the eccentricity and the radius of the left ventricle, so that as that septum bows, you can see this change of the left ventricle from this nice circular shape to more of that D shape that's suggested that the right ventricular pressure is pushing on the left ventricle. So this is another, this is a healthy rat cardiac MRI again. As we can see, the right ventricles kind of and the left ventricle are synced up really nicely. They're beating together, and then this left ventricle is maintaining this nice round shape. And then at just 3 weeks later, this is these are actually MRIs that were just done a couple weeks ago. We can see that the right ventricle has increased significantly in size, and that this the RV and the LV aren't really moving together as well as they were before. So there's a little bit of kind of decoupling of the right ventricle and the left ventricular contractility. And then here we're seeing a little bit of the septal bounce, which is indicative again of right ventricular pressure overload, and that the septum is kind of moving into the left ventricle every time the right ventricle contracts. So this is, you know, if I saw this in a patient, I would definitely be concerned. And these are, again, just rats at 3 weeks of Sugen hypoxia. We're planning on taking this out to a 9-week time point. So putting the rats in, they return back to sea level or normoxia for another 6 weeks. And then we're going to do a final set of cardiac MRIs to see how much further the right ventricular morphology and function changes at that point. So in this rat Sugen hypoxia model, I earlier did studies to say, so, why do we care about these cardiac macrophages? We're characterizing them, we're looking at these different surface markers and what's increasing, but are they actually relevant? So how we studied that was to use clodronate, which in a liposomal form gets taken up by professional phagocytes like macrophages and dendritic cells and results in the death or the apoptosis of those cells. So we administered clodronate liposomes to rats in Sugen hypoxia. So we gave it on the first day prior to them receiving Sugen and being placed in hypoxia, and then we administered it weekly after that to maintain what we hoped was the macrophage depletion. And so this is an example of, this is actually mouse heart tissue, but this is an example of 1 week after clodronate, staining for CD206, which is another macrophage marker, and MERTK. And so at 1 week after clodronate, there's much fewer identifiable macrophages in this stain. So in this set of experiments on using clodronate for macrophage depletion and Sugen hypoxia, we saw that the Sugen hypoxia plus just vehicle-treated rats and the Sugen hypoxia clodronated-treated rats all developed pretty severe pulmonary hypertension with RVSP in the 60s, and that both of these populations also developed pretty severe right ventricular hypertrophy and elevated Fulton index, which is so unchanged compared to what we know as the control conditions of just Sugen hypoxia without any clodronate or PBS liposome treatment. And then we saw that in the extracellular matrix component or the right ventricular matrosome of these rats, the macrophage depletion significantly altered the right ventricular matrosome, so that in control rats, there was, so control meaning they got Sugen, but then got just PBS liposomes and not the macrophage removal. There was higher levels of all of these collagens as well as these other extracellular matrix proteins, whereas in the clodronate or the macrophage depleted rats, the extracellular matrix compartment had much lower levels of these multiple types of collagens and fibrillins. And so does that affect the right ventricular function? So we found that using intracardiac catheterization, we could measure the contractility between the clodronate versus the PBS treated rats. Again, I'm losing my significance markers for some reason, but there was a significant increase in the contractility in the clodronate macrophage depleted rats in the right ventricle. And then also there was an improvement in the right ventricular PA coupling. So, so in these experiments, we showed that removing the macrophages did matter, and that in this severe model of Sugen hypoxia PH, we actually improved the function and the contractility of the right ventricle and improved the RVPA coupling. So summarizing all of these experiments, experimental pulmonary hypertension stimulates, experimental pH stimulus increases certain subsets of right ventricular macrophages. So whether you kind of separate them by this IM1, IM3 phenotype or by CCR2, experimental bleomycin, at least in our hands, seem to increase these tissue resident CD163 macrophages. And then macrophage depletion and Sugen hypoxia pH significantly changed the RV extracellular matrix, improved RV contractility, and improved RVPA coupling. So now the question is, what are all these subsets actually doing? So what are these macrophages programmed to do? And by what mechanisms are they actually affecting the right ventricular extracellular matrix or affecting the right ventricular contractility? So that essentially needs further study, and that's what some of my future experiments are going to be focused on. So my future directions are going to be looking at the complement system and interactions with macrophages in the right ventricle. And I know we've actually had a couple of complement talks in this forum recently, and I think there's even more coming up, but just to review, there's 3 pathways of complement: classical, lectin, and alternative pathway, and they all converge on making the C3 convertase, which cleaves C3 into a product called C3A. That by itself can interact with receptors like the C3AR1 receptor, which is present on macrophages and other inflammatory cells, and cause inflammation in the resident tissue. And then this process also creates C3B, which is a molecule that functions to opsonize cells or things that the resident cells are going to recognize as foreign. So in cancer, this is relevant. For example, it's also relevant in kind of our host immune system. So this helps professional macrophages or professional phagocytes like macrophages and dendritic cells to recognize what needs to be opsonized. So these are 2 ways that the complement system likely interacts with macrophages. And then this kind of further, more terminal pathway is making the C5 convertase, which cleaves C5 into C5A as well as C5B. C5A also interacts with C5A receptors, which are present on macrophages and other inflammatory cells. But then C5B goes on to make this MAC-ATAX complex, which I think we've heard about as kind of the terminal complex that helps with cell lysis and activation, and is really relevant in our immune cell host defenses against bacteria and other pathogens. So getting back to why complement might be relevant for pulmonary hypertension, in humans, what's been studied a lot is the serum of PAH patients. So we know that C3 complement is increased in the serum of PAH patients. And in this study, which looked at the altered proteins in serum of IPAH patients, actually complement had the biggest fold change in terms of the ratio of IPAH compared to controls, at almost 6-fold difference. And then in experimental models of pulmonary hypertension as well, there has been shown to be increased complement pathway signaling. So in in this study, it was a study of monocrotaline pulmonary hypertension, which I didn't really talk about, but it's another severe PH experimental model, as well as the Sugen hypoxia rat model, looking at the differentially expressed genes and the enrichment of pathways. Again, complement comes up as one of the pathways that is significantly changed in the right ventricle of the pulmonary hypertensive animals. And then they validated that just with traditional RT-PCR by looking at one complement gene, which is C5AR, which is the receptor for C5. And again, this the relative expression in the right ventricle of Sugen, hypoxia, or monocrotaline animals was significantly increased to controls in the right ventricle. And then this increase is not seen, or it's not significant in the left ventricle. And then the C3A and C3AR signaling pathway seems to be relevant in regulating cardiac remodeling and right ventricular fibrosis and failure. And in this paper recently published in 2022, using a mouse model of PA constriction, there are multiple markers other than just RV and diastolic pressure that were improved in C3 knockout mice. And they also used a few different transgenic mice that were deficient in C3 or in liver and heart-specific C3. So in all of those animals, as well as in animals treated with C3 antagonists, they showed that there was decreased fibrosis and decreased changes in RV. So in this one figure example in their paper, in these C3 knockout mice, you can see on trichrome staining that there's much less fibrosis in the RV. So this sets up our studies. So we're currently doing staining for complement and markers of complement activation in both human and the animal RV and pH. So this is one stain of C3A and DAPI in the human RV in a pulmonary hypertensive autopsy case. So you can see that there's this whole region here where there's a lot of C3A lighting up. And then the interaction of the complement system with macrophages, as I said, is through the receptors that we know are expressed on the macrophage surface. So the rat RB macrophage does express C3AR in high quantities. So in the flow cytometry studies that I've done on rat macrophages, again, we identify the CCR2 positive population of macrophages, and the predominant, these CCR2 positive macrophages do predominantly express C3A receptor. So over 90% of CCR2 macrophages are expressing C3A receptor. So that all kind of hints at this interaction between complement and macrophages in the right ventricle. This is showing that in the studies that I've done of kind of all C3AR myeloid cells in the rat heart, the majority of these cells are macrophages. So whether it's in the right ventricle or left ventricle, over 60% of C3AR myeloid cells are from the macrophage lineage. So my hypothesis for my future studies are that alternative complement pathway activation, so proteins and RNA expression of C3, C3A, CFB, CFD, and C5A are interacting with the C3A receptors and C5A receptors on macrophages, and that these then lead to activation of the macrophage and secretion of pro-inflammatory cytokines that over time can lead to RB failure, such as through artery dilation, fibrosis, or decreased myocyte contractility. And I'm planning to treat animals with C3AR antagonists to see whether blocking this macrophage activation by complement can rescue or prevent some of these changes in the RB. So we're currently doing a lot of projects to gather preliminary data on this next direction. So we're doing Western blotting for C3, C3AR1, and control on pH rats. We've done RT-PCR to kind of confirm the published findings that there's increased C3 and other complement markers in the right ventricle. As I've shown, we're doing flow cytometry to identify the C3AR, C5AR receptor expressing macrophages in the rat heart. We're doing both rat and human staining right now, and the human, the HIMSR core is helping us do multiplex immunohistochemistry staining, where we're going to overlay those areas where there's increased C3A with different macrophage markers to see if they co-localize and if there's more complement around areas of specific types of macrophages. And then we're also going to do fibrosis assays, just trichrome staining on the serial sections of the same human hearts that we're doing the multiplex staining on, to see if those areas of the C3A and increased complement activation overlay or co-localize with the fibrosis in the right heart. So that's all currently in progress, and hopefully we'll have updates in the next several months about this new project in complement and right ventricular macrophages. So again, hopefully I have shown you the importance of the right ventricle in pulmonary hypertension, and I've shared with you what I know about how to characterize macrophages and their role in the right ventricle and pulmonary hypertension, and hopefully got you excited about this possible new direction of the contribution of complement activation of RB macrophages to right ventricular failure and pulmonary hypertension. So these are, I have a lot of acknowledgments. So Kurt Stenmark is my primary mentor, and the CDP lab, everyone there has helped me with a lot of these preliminary experiments. Tim Lamb at National Jewish is my career mentor. The Ronald Bagnozzi lab has partnered with us on a lot of our studies on the reporter mice with CCR2. And then the animal imaging shared resource, Jenna Steiner and Natalie Surkova started these initial cardiac MRI scans in the rats, which are really beautiful and exciting to look at. And as I mentioned, the HIMSR core is helping us do the immunohistochemistry multiplex staining. My research advisory committee and National Jewish Macrophage Labs are my go-to resources for all things about phenotyping and function of macrophages. So I'm ready for any questions.
Do we have any questions? And I'm going to ask you to please repeat the questions so that the people on Zoom can hear.
Sure.
Go first.
Neil?
Sue, great talk. So you may or may not be familiar from the acute lung injury literature, the IT bleomycin gets used in that model as well. And people that study fibrosis notice that over time, changes in the lung tend to resolve. And so IP bleomycin has been used. I was curious if that has been used for pH studies, and if that would actually cause some RV dysfunction and sort of provide that chronicity that you see with pH. That's one question. The second is that what's your general sense of, just in thinking about macrophage populations that are either resident or recruited, with these different injury models that have different time courses of severity and sort of peak inflammation and resolution? Like, what do you think is there a right time to study, these immune cell populations, or do you think it just is, you have to look at the kinetics for all of them and sort of look at the course that way?
Right. So the first question was whether IP bleomycin is used as pH, and I do know that there are labs that do that as a pH model. I don't know if the pH resolves. That's actually a really good question. It's not, even the IT bleo isn't a model that I've used very often. So I'll have to look that up and get back to you. And then in terms of when to study macrophages, I think this is like the really tricky question. And I think my work has really kind of been characterizing pH, characterizing what happens with macrophages and pH. But I think that the next step is really to hone down on understanding one model really well and to study that time course really well. So honing in on the right model that really, really has all the features of RV failure that I'm interested in studying, the rat suturing hypoxia model. So I would like to eventually understand what happens in early pH, whether those macrophages are needed for that first adaptation in the right ventricle to, acutely increase its contractility in response to the pH. And then what happens as the pH goes on later on, if you intervene or if you remove macrophages at a later time point, is that better for the rat or not? So that's definitely something that I think needs to be studied carefully and with with nuance. And I think that the cardiac MRI or echoes are these kind of functional methods of catching exactly where in the disease process it is. So I think even at 3 weeks, it's a little bit kind of already, I would say, you've already entered into a phase of maybe moderate or kind of compensated RV failure if you're already seeing some septal bowing. So being able to do longitudinal studies of animals and then sacrificing them and looking at the macrophages at those time points before they developed right ventricular failure, and then to be able to follow some animals until they have developed clear evidence of right ventricular failure and looking at the differences in the macrophages and the complement in those two different time points. I think that's where I'm hoping to start. Fernando?
So very nice work, really wonderful. I think that the macrophage is one of those cells that can really switch metabolic reprogramming depending on its immune phenotyping and activation. And I know one of your slides showed that there are a lot more on the oxidative phosphorylation phase, so they're probably more glycolytic macrophages. Have you looked at metabolic phenotyping of the cells, and maybe how does that contribute some of the changes you see on pH? So I know there's a very rich literature about myocyte metabolic reprogramming and pulmonary vascular endothelial cells metabolic reprogramming, and I'm just seeing how that flavor looks like.
I have not yet. So I think what I'm hoping to do is, now that we're focusing on the rat and we're able to do these longitudinal studies, is to find ways of isolating macrophages in a way that we can study them for RNA sequencing or from metabolic programming. Since CD68, which is the macrophage marker, is unfortunately intracellular, flow cytometry requires fixing those cells. So what I'm hoping to do is to use magnet isolation beads to separate out those macrophages so that I can do these studies of the rat macrophages.
I was surprised by the clodronate results that you got, because you weren't seeing changes with clodronate in RV function or RV hypertrophy, right? If I saw that
Right.
correct. So you might know Kurt and Maria Frid,
Yeah.
like in 06, I think,
Mm-hmm.
did the clodronate in the bovine and the rat model, hypoxia model.
Mm-hmm.
I can't remember what they showed for RV changes or RV pressure and hypertrophy and all that, but I know they showed profound changes in the pulmonary vasculature.
Mm-hmm. And there and in whatever time course they studied it in, those animals were protected from pH. So they
That's what I thought.
did not develop the they didn't develop the perivascular inflammation and the kind of intimal thickening and all the changes that are characteristic of pH. So so I'm not sure exactly what we did differently where these rats still developed pH and I was able to capture that, and then what they did that prevented that early initiation of pulmonary hypertension.
I have another question. I didn't know resident max actually regulate conductance. So clodronate is pretty much a sledgehammer, right?
Mm-hmm.
How come we didn't see change? Would that affect the resident max? And if so, wouldn't that affect cardiac function?
Yeah, it should. Maybe, just to repeat some things for the Zoom, the question was, since clodronate's kind of this like very non-specific way of depleting macrophages, would it also affect all those other, resident macrophages that I showed at the beginning that were regulating conduction and, other things? And I think that is a good question. I don't know at what doses it penetrates and like gets to what specific macrophage populations. I think that is the problem with clodronate, is that we, and at the time that we were doing those clodronate studies, there was probably like 4 flow cytometry markers available for use in rats. So it was very hard to characterize the macrophages at that time. But it would be definitely interesting, I think, to do a clodronate study and to really carefully use tissue immunostaining to see where the macrophages are actually being depleted.
There's some anecdotal suggestion that clodronate is less efficient at depleting resident max than circulating. I think the paper from the Narendorf group that did that conductance, they used a DTA type model to really target the resident cells. So it could be that too.
Right. Yeah. Yeah, it was a very cool, like a DTA. It was like a light activated depletion model. So when they shined the light on those animals, it depleted those macrophages and it caused AV block. So it was very cool.
Any other questions?
Neil, you had your hand up for a second.
I was just gonna, so our experience in the lung is that clodronate given systemically doesn't deplete alveolar macrophages, so the ones that are truly on that side of the barrier.
Okay.
Obviously with repetitive doses, you're actually causing some injury, so you may see some barrier disruption over time, but at least in the acute setting, we didn't used to see that depletion happen.
Thank you.